From the JAMA Network, this is JAMA Oncology Author Interviews. Conversations with authors exploring the latest clinical research, reviews, and opinions featured in JAMA Oncology.
Hello, and welcome to our listeners around the world. You are listening to JAMA Oncology Author Interviews. Thanks for joining us. I am Vivek Subbiah, medical oncologist and chief of early phase drug development at the Sarah Cannon Research Institute, Nashville, Tennessee. And I'm also the JAMA Oncology podcast host for today and the web editor for JAMA Oncology. Joining us today is Dr. Jiyoung from the NYU Grossman School of Medicine. She's the lead author of the recent publication in JAMA Oncology of Oral Bacterial and Fungal Microbiome and Subsequent Risk for Pancreatic Cancer, a population-based nested case control study that spans over 122,000 individuals. The research reveals compelling evidence that the oral microbiome, both bacterial and fungal, may serve as a powerful, non-invasive biomarker for identifying high-risk individuals with high risk of pancreatic cancer. Dr. Jiyoung, thank you so much for being here. Please, can you go ahead and introduce yourself?
Thank you for having me today. My name is Jiyoung. I'm a professor at New York University Grossman School of Medicine and also associate director at NYU Perlmutter Cancer Center. By training, I'm a cancer epidemiologist. My research focuses on the human microbiome and cancer.
Thank you so much. Again, congratulations on this fantastic publication. It was fascinating for me to read the paper to show how periodontal pathogens and fungal species, specifically like Candida tropicalis, are linked to cancer risk. So let's get started here. So what inspired you to investigate the link between oral microbiota and pancreatic cancer? And how did you decide to include both bacterial and fungal communities in your study?
Pancreatic cancer is one of the deadliest cancers. Right now, we don't have a good screening test and treatment options are limited. So we only know a few risk factors like a family history, smoking, and obesity. So finding new modifiable risk factors is very important because it can help us prevent this disease. About 10 years ago, our group reported that people with a poor oral health seems to have a greater risk of pancreatic cancer development. We suspected that this could be something to do oral bacteria. More recently, animal studies by other groups showed that bacteria from the mouth can actually travel through saliva into the pancreas. But we didn't know what exact species of bacteria and fungi might be involved in the pancreas carcinogenesis. So therefore, we conducted this large human study to examine the oral microbiome, including whole bacteria and fungal profile in the mouth, and to see which bacteria and fungal taxa are associated with the subsequent development of pancreatic cancer.
So you talk about this risk-based score based on specifically the microbial risk score, MRS. So here in your study, you note that you found a greater than threefold increase in pancreatic cancer risk based on the microbial risk score, MRS. So for our broad audience, can you describe, one, what is this microbial risk score, and how do you envision this score being used in clinical practice or screening programs?
So we identified a total 27 individual bacterial and fungal species significantly associated with the pancreatic cancer development. Some are positively associated, increased risk, while others were inversely associated. So altogether, we summarized the effect of these total 27 microbial species by creating a microbial risk score that combined each species' effect size and abundances. So using this approach, we found individuals with a greater score had a 3.5 times increased the risk of developing pancreatic cancer. This could serve as an important risk stratification tool in healthy populations. If validated, so oral microbiome profiling could serve as a non-invasive biomarker to identify individuals at elevated risk who might benefit from enhanced screening surveillance, which is especially important given that the lack of effective early detection method for pancreatic cancer.
You specifically mentioned this Candida tropicalis that was associated with increased risk, whereas Candida albicans showed a protective association. Could you elaborate on the biological mechanisms that might explain these contrasting roles?
Sure, great question. We showed for the first time that oral fungi, particularly Candida, may also play a role in pancreatic cancer. Epidemiologically, people with a frequent oral Candida infection, so-called candiditis, appear to have a greater risk of developing pancreatic cancer. So Candida is known to be an inflammatory fungi infecting multiple body sites, including the mouth. And at the fungal genus level, we found the Candida genus was linked to be associated with increased risk. However, when we examined this data into at species level, about 30% of the sequences remained unidentified, so unclassified. So we don't yet to know exact species level involved. Since fungal genetic reference database is relatively new and yet still incomplete and continuing to evolve, species level finding might be interpreted with caution. But interestingly, we did identify a Candida species in human pancreatic tumor tissue, matched the sequence from oral samples, suggesting a possible connection between oral fungi and the pancreas. So going forward, we need more mechanistic research to understand exactly how these fungal species might contribute to pancreas cancer development.
Thank you, Ann. So, given the strong association with periodontal pathogens like Porphyromonas gingivalis, what role do you think oral hygiene and periodontal disease management could play in cancer prevention strategies?
So among the oral bacteria associated with the risk, three bacterial species are well-known periodontal pathogens, including P. gingivalis, which cause periodontal disease. This is consistent with our previous research findings linking poor oral health to pancreatic cancer development. So these findings suggest that maintaining good oral hygiene through regular brushing, flossing, and dental care is critical. This is a modifiable risk factor for pancreatic cancer prevention. This is also aligned with the broader public health messages, such as the US Surgeon General's report on oral health, which emphasizes that oral health has important effects on overall systemic health, including cancer.
So, how did you ensure the reliability of the oral microbiome data over time, especially considering the one time sample collection?
We used a prospective study design, so we were able to examine oral microbiome profiles in healthy individuals and then track which oral microbe were associated with the subsequent development of pancreatic cancer. So this unique study design allowed us to assess a temporal sequence, which is critical because most microbiome studies are cross-sectional, which the microbiome profiles are already influenced by the presence of cancer. So here, a key assumption is an individual's microbiome is reasonably stable over time, unless a major event occurs. So in a separate pilot study, we collected repeated oral samples over years, and we showed that oral microbiome is surprisingly stable over years. Each person tended to maintain their own unique microbiome profile, which is distinct from that of others. We think our findings are fairly robust. We analyzed two large independent cohort populations, and results are remarkably consistent across both populations.
I see. Again, that is very interesting how you compare and contrasted to validate your research and make it more reliable. So, were there any surprising findings in the fungal microbiome analysis that challenged your initial hypothesis or opened new research directions?
Sure, we are surprised to find that pancreas is full of bacteria and fungi. We think inside of our body are quite sterile, but which is not. Our next question is what these bacteria in the pancreas do. We are currently investigating whether the composition of the pancreas tumor microbiome can influence cancer treatment response and long-term survival in pancreas cancer patients.
Wonderful. So, what are the next steps for translating these findings into actionable clinical interventions or further research, especially in multiple populations and diverse population beyond those studies in the CPS2 and PLCO here?
So the PLCO and CPS2 cohorts are excellent resources because they provide large scale, long-term follow-up data. However, nearly all participants are white. Studies suggest that microbiome profiles vary across different race and ethnicities. So it is really critical to validate our findings in populations, non-white populations, including Asian Americans, Blacks, Hispanic individuals. So it is also important to note that our study shows associations, but it does not prove causality. So we need additional mechanistic studies, additionally, ideally randomized clinical trials to determine whether this microbe will play a direct role in pancreas cancer development.
Thank you so much. So, again, in summary and conclusion, right, what are your closing thoughts for our broad audience of JAMA Oncology? This is fascinating research. And also, in your closing thoughts, please share how do you envision this being implemented in clinical practice or screening programs in the real world?
Well, so this is really a first study provided concrete scientific evidence of the role of oral bacteria and oral fungi in relation to the pancreatic cancer development. So as I mentioned here, this will be a useful screening tool, risk stratification tool. Knowing the oral bacteria can help us to figure out who are at the risk and subsequent development of pancreatic cancer and who might deserve more rigorous surveillance in terms of pancreatic cancer prevention.
Thank you so much. I'm Vivek Subbiah, and I've been speaking with Dr. Jiyoung about oral bacterial and fungal microbiome and subsequent risk for pancreatic cancer. You can find a link to this article in this episode's description. To follow this and other JAMA Network podcasts, please visit us online at jamanetworkaudio.com or search for JAMA Network wherever you get your podcasts. This episode was produced by Daniel Morrow at the JAMA Network. Thanks for listening.
This content is protected by copyright by the American Medical Association with all rights reserved, including those for text and data mining, AI training, and similar technologies.